This is a production of Cornell University.
Welcome you to the Restoration Ecology 2017 cohort's presentation of their class project. And one of the objects of the project is to expose the students to experiential learning. You know, a situation where, you know, you try once, you fail, you try twice, you fail, and then by the third or fourth time you finally get it figured out how you actually should do it. And what looks really good on a blackboard or whiteboard in a classroom situation changes very rapidly when you are in hip waders and you are in mud maybe up to your knees. You say, well, maybe I can only do one of these sample sites, or maybe I can only collect one kind of sample. What we're looking at right now is one of many drone overflights that we did of, it's called Treman Lake, or Lake Treman. It's more like Treman Lake and Marsh Complex. This is the dam, and whoa. So we were able to do a number of very interesting things over the past several months, including these drone overflights, and we spent a lot of time canoeing around and sampling the vegetation and collecting soil samples and so forth. So before we actually get started, I'm going to encourage everyone to grab a bite to eat if you haven't already done so. Anybody still need lunch? Okay, okay. This is the class. Wow. They look a little bit different than they do at 8:30 on a Tuesday morning. It's really good to see everyone, all of you and all of them. And check this out. Here are two members of the class standing in the middle of a little peninsula on the marsh. So I'm going to turn things over to class members right now, and Audrey.
Wonderful.
Oh, okay. Audrey was our TA this year, and did a really good job of keeping me honest and making sure that we didn't leave things behind. We didn't lose any students. So, Audrey, do you want to do your introduction and then?
Thank you all very much for coming today. This is exciting. We spent a full semester working together a lot, and it's wonderful to see it come together. So thank you. I'd like to begin with some history. So starting around 1930, the Civilian Conservation Corps put in a dam at what was Buttermilk Creek, creating Lake Treman. And so slowly over the years, the area has been filling with sediment, just like the southern ends of the Finger Lakes. They've been filling with sediment ever since the glaciers. And so Lake Treman is really a community asset. It provides ecosystem services like sediment mitigation. And so,
please speak louder.
Of course. The sediment is trapped via the dam, and so it doesn't go down into the swimming area or Lake Cayuga. This is really helpful to the community. Over time, the area has changed from glacial to creek to lake, and now we see it slowly transitioning into a wetland marsh. The question now is what we want the system to do, what it can do for us, and what we would like. So when talking with the New York State's Park Department, we decided to address a couple important issues for managing the area. First and foremost, we wanted to estimate the depth of the water and the volume of sediment behind the dam. To do that, we outfitted a canoe with a handmade bathymetry device, and we put on waders, and we used 1989 maps to do these calculations. You'll see some of these calculations later. It's pretty exciting. We also wanted to address the invasives on the site. And so the state was looking into getting funding for this, and we wanted to help, so we put on waders, we grabbed a GPS, we started mapping where the Phragmites was and where the knotweed was. In doing this, we discovered that there was a large diversity of plants that we didn't think would be there because it looked like a cattail marsh. We also wanted to address environmental and human health. And so we sampled for bacterial contaminants, that would be upstream and downstream of the dam, to see if there are any differences and what the results would be. We also looked at heavy metals in soils and methane in the sediment. And at the end of the day, we really wanted to recognize the ecosystem services and the functions that the site is currently providing, and we want to provide realistic options that will help these functions continue. And so with help from community experts, some of which are here today, we've done a lot of great work, and we're excited to present it. So with that in mind, thank you all for coming, and I'll hand it over to Meg for the presentation outline.
All right, thank you all for coming. Exciting stuff. This was our entire semester, so a lot of work and time went into this for sure. These are all the general things we're going to be going over today in greater detail than Audrey mentioned. As Audrey mentioned, the lake was built in 1930 by a dam by the CCC. For many years, it existed as a lake, and it was important to the beauty of the area and the beauty of the recreation facility. Also important to keeping sediment out of the swimming pool downstream. But over time, it filled up with sediment, 16 acres worth about, and as it is now, it looks nothing like what it used to. This is a historical map. It used to be a stream, and it was turned into a lake by the dam. Within 80 some years, it is now a wetland, and what we want to know is the wetland really that bad, or does it provide similar ecosystem services as the lake? And should it be changed, reverted back to a lake, or kept as a wetland? Okay, so some site characteristics. These are the positive aspects of the marsh, not all the negative aspects. It prevents sediment from flowing into the swimming area, and with that sediment, also fecal coliforms. It provides an ecosystem for a variety of plant species and animal species, and it provides a different aesthetic than the lake. You saw the video, it is quite beautiful. But what we want to know is if it's worthwhile in keeping. I'll hand it off to Tanvi, who is on the mapping team.
I prefer not to use the mic. Can everyone hear me if I don't? Okay. General overview is that Lake Treman doesn't exist in limbo. There's a pretty large watershed contributing flow into it. The lake here is represented by that tiny set of black points in the top left corner, and the watershed itself is very large. So this is a map of a digital elevation model. Some very amazing people have allowed us to have a 1 meter resolution DEM, which is what we used along with curve number calculations to delineate the watershed. So this is the DEM displaying elevation. And then this is a layer representing curve numbers. So high curve numbers mean that there's low permeability in that soil, and vice versa for low curve numbers. And this is a function of both soil type and land use type. So this was used in conjunction with the DEM to delineate the watershed. This gives us a little bit more information about what kind of runoff Lake Treman is receiving. So in yellow is agricultural land use, and that kind of represents the hot spots where we might expect problematic runoff to be contributing to the lake. Using the hydrological models from the DEM and storm runoff, we could model the general flow of streams in the area. And if you zoom in further, this coincided pretty accurately with the path of water up until we get to Lake Treman, where there's been a slight divergence, as we would expect. So using all of that data and NRCC storm information, we calculated runoff for a 24-hour storm for various frequencies. So this is an example of a frequency duration curve for a 1-year storm, a storm of intensity that we can expect to see once every year. We did this for various frequencies, up to a storm that you would expect to see once every 500 years. And these are the corresponding runoff rates that we would expect to see at the dam point. It's commonly accepted practice to model climate change in the year 2050 by multiplying existing precipitation rates by a factor of 1.5. So these are the flows that we could expect to see 50 years from now, or less than 50 years because it's 2017. And then I'll hand it over to Kevin to talk about comparison with historic maps.
Yeah, so in this 1938 picture, you can see the lake perfectly as a lake, and there's no marsh included. But as time progresses, you begin to see the encroachment of the marsh, which currently stands at 10.89 acres of the original about 13 to 16 acre footprint of Lake Treman, original Lake Treman.
So then, using those maps, which Susan Hoskins directed us to, we could see, we could look more closely at how the extent of the lake has changed. So this is a more modern representation, which is still slightly bigger than the current extent. This is from the 1950s. You can see that the lake displayed is significantly larger, with emerging wetland in the south, depicted by those dots. I don't know if you can see them currently, but they're emerging. And then this is from 1898, before the dam was constructed. We're not completely sure about how accurate this is because of the equipment that they had in the 1800s, but it's the best estimate that we have. And you can see here that the lake falls in this area, and the elevation ranges from 940 feet to 920 feet. And we use that information to determine the slope while modeling the historic elevation of the stream. So this is just a side-by-side comparison. And so our DEM shows us the elevation of the surface of water, but doesn't tell us what the elevation of the underlying sediment is like. So Dustin and Jonas and I went out in a canoe with a bathymetry device and got all these data points representing the depth of water at different points in the lake. And we could interpolate those to come up with the general surface of sediment underwater. So we could subtract the volume of water from the current lake. But then we also had to model the historic stream because surprisingly there were no DEMs in the 1800s. So we did that. I can talk more about the methodology because it was slightly involved, but we basically just raised everything to the highest elevation, sloped it, and then dug out the channel.
What is a DEM?
A DEM is a digital elevation model, so it's basically just topo lines at a very high resolution. So then these are our estimates for the current volume of water in the lake. Acre feet, it might be an easier measure to think about than meter cubed. So if you thought of a 63 acre area, it would be raised to like 1 foot in depth, and an acre is approximately 1 football field in size. So the annual deposition is pretty high, and we could expect the sediment to fill up the lake in about 30 years if current conditions remain the same. To reduce deposition rate, there are certain hotspots of erosion upstream of the lake that we could address. We could also construct a channel adjacent to the wetland to allow the water to have a higher velocity, meaning that it would be depositing less sediment. And Kevin has identified areas of erosion hotspots.
Yeah, based on the runoff coefficients and the land use, we can see certain areas where agricultural land and bare agricultural land contributes mostly, or we believe that contributes mostly to the deposition of sediment. And so it's these critical areas upstream of the dam that we believe that the state or somebody should really look into more depth about controlling the rate of sedimentation. So as a step to maybe elongate the amount of time we have until it needs to be dredged or some other measure to control for the deposition of sediment.
So then stream bank analysis was conducted at various sites upstream of the lake to determine what the channel looks like without human influence or without too much human influence. So we did this manually with levels and meter sticks, and we found a general trapezoidal cross-sectional shape to the channel upstream of the area. And this is just 2 more from other sites. So based on the cross-sectional area and the slope of the stream, we determined that using the Roskin classification for streams, which is kind of like both hydrological and ecological, it falls between C and B. So kind of an intermediate slope, liable to changes in meander shape. The services provided by the current system with the wetland as it is are that it's potentially treating agricultural runoff. So we do have some water samples that we tested, but extensive water sampling could give us a better idea of what this is. It serves as a flood control system currently because it is essentially a retention basin. It allows us to control flow downstream with the boards, as the park currently does, and it provides a unique wetland habitat. The hydrological risks are that there's rapid deposition happening, which leads to a reduction in basin capacity. So if we get used to it serving as a flood retention basin, that might change in time if we don't control for erosion. Additionally, it will mean that the wetland gets flooded less frequently, and that can be a habitat change. So our recommendations are to perhaps construct a channel adjacent to the wetland from the hydrological perspective. A trapezoidal cross-section is what we recommend based on upstream characteristics. And to avoid erosion, we have more detailed measurements about what the side slope ratio should be. To avoid erosion, we recommend that the bed be lined with cobbles and shingles, so basically layered rocks, as you would naturally see in such a stream, because they have a very high roughness coefficient, here expressed as Manning's N, meaning that they're more resistant to erosion. But even then, for a channel to carry the discharge from a 2-year storm without any erosion, it would have to be really, really big. The reason for that is because erosion naturally occurs during such storms. I'm sure no one has seen a stream during a very heavy storm not have some amount of sediment suspended in it. So there are 2 ways to perhaps combat that. One is through a wetland stream complex, where we direct flow from the wetland into the channel through a permeable embankment. So we would essentially have a wetland on one side and a channel adjacent to it, and they're sharing flow underneath the surface if we make this embankment out of a permeable material. And that also provides 2 different habitats. It allows for a more riparian habitat on one side and a wetland mosaic on the other side. I'll now hand it over to the methane team.
Hi everybody. So as we've already talked about a bunch, there is a lot of sediment that has accumulated behind the dam, and that has, because of the texture of that sediment and the amount of water behind it, has led to methanogens producing a lot of methane. That's a very anoxic environment underneath the dam. And that means that, well, greenhouse gases would normally be produced. Methane is produced in this case instead of CO2, and methane is a much more powerful greenhouse gas. And the water in the reservoir that the dam has created also acts as a barrier, not allowing the methane that is created through the sediment and the microbes in the sediment to be released. It keeps the bubbles down from the pressure. This is important to our recommendations for the site because it depends on whether the stakeholders involved care about this as a potential source of greenhouse gases. If there's a lot of methane that could be released, it would probably not be wise to allow the water to drain out.
So we wanted to test how much methane was being produced in the wetland. These are the sites that we dredged from. We used an Ekman dredge, and we took samples throughout around the dam. I just wanted to point out that these areas we think have more running water, and these areas have more stagnant water, where 1, 2, and 5 were taken. So from each sample, we took 20 grams and put them into airtight mason jars. Then we pumped out the oxygen and pumped in nitrogen to create an anoxic environment for methane to be produced. We incubated it for a week and then used gas chromatography to measure how much methane was in the mason jars after that 1 week. Then we enriched the samples with 2 grams of dry cattails each to add some more carbon for the methanogens. We again took out the oxygen, replaced it with nitrogen, incubated it for 1 week, and then tested how much methane was produced in that week. And Jonas is going to talk about the results.
I'm just going to use this one. So I'll run through some of the results real quick. So we have the raw samples here, and then we have, as Jasmine was talking about, the enriched samples where we had 2 grams of cattails. Just to make it a bit more clear, we have here. So this is the methane produced in each of the samplings down here, and we have an average. So as we expected, we see a higher methane production in the enriched samples because we added a substrate there. So that was kind of what we expected. And as Jasmine was saying as well, sample 3 and 4, where we expect more running water running through, we wouldn't expect like high methane production there. It was these 2 samples here because we expected all the water to come through here. So a low sedimentation there, or where we have 1, 2, and 5, where we expect there to be more sediment depositing here. So what we wanted to do was to calculate the methane produced by the lake and the wetland area. So we found some numbers for the sediment density, and we made the assumption that methane is only produced in the top 10 centimeters of the lake, because below that we don't really have any carbon source. By the help of Joey Abbott of the Department of Natural Resources, we calculated this, and we got a methane production per year to be 14 to 27 kilograms a year. To put this in perspective, we looked at this guy, and a cow can produce around 58 to 144 kilograms of methane a year. So that means that 1 cow produced 2 to 10 times as much methane a year than the lake/wetlands. Again, consulting with Joey Abbott, we looked at the global wetland emission of methane, and these numbers are actually pretty low when you look at the a standard or the average wetland methane production. So our recommendation would definitely, the methane production in the wetland/lake wouldn't be that big of a concern. It's always a concern, of course, but not as much as it could be. I'll hand it over to Benthic Group.
Hi guys. So we went out on September 30th and we just went to collect benthic invertebrates. We did 8 we did 8 downstream sites and 3 sites in the lake, but we were only able to get invertebrates from one of those sites in the lake, which was interesting to say the least. And that's some of us kicking into the nets. We went out again on October 13th, just because we wanted more sampling from upstream. So we went to 4 more sites off of Comfort Road, and then we did some seining with Jeremy, who's in the crowd right now. He was very helpful. So this is just more of us in the field. It was an interesting time. I learned a lot about inverts. So we collected 199 invertebrate species were identified by family. 16 families of invertebrates were identified, and these are 4 drawings of what they were. So yeah, like Tipulinae, Heptaginae, and then we did 4 species of fish were identified. So we wanted to like make sense of what all of this means. So we did an HBMI, and HBMIs are essentially an invertebrate's ability to withstand like pollution. So like the lower your HBMI, the less resistant you are to like pollution. So you kind of want your numbers to be about as close to 0 as possible, because that kind of is indicative that your stream is not as polluted. So yeah, so we did that calculation, and so we divided it in terms of upstream pool, downstream pool, upstream riffle, downstream riffle, and our entire site is about a 4, so it's moderately polluted. I don't think it's too much to be worried about. One thing that was interesting was the upstream pool is only about a 2.22, and we didn't get enough data to see if that's significant, so more testing should be done to see like what happened, if that's actually something that's less polluted, I guess. And so then we collected all this invertebrates, and we could put it on a pie chart, but we wanted to figure out a way to, what does this mean? How can we compare these two? Like, okay, like D1 riffle has all these species, and in D1 pool we only had like this one species of invertebrates. So like, how can we compare these two? So we decided to do diversity indices, and one of them was the Simpsons index. So we looked at downstream pool, downstream riffle, and then the entire downstream system and the upstream system. And our conclusions were downstream pools were more diverse than riffles, and downstream in general was more diverse than upstream. And then we did another diversity index called Shannon-Wiener, and so we'll essentially be looking at H, and this one kind of said the opposite thing. It said downstream riffles are more diverse than pools, and upstream is more diverse than downstream. And I think, or I suspect that it's probably because of how they're calculated. If you notice in the denominator for Simpson index, it kind of doesn't give as much weight to, if we found one species, it would be 1 minus 1, which would make the denominator 0. So I suspect that that's probably why our two indices don't agree on that front. And then I'll turn it over to Dustin.
Thanks for that. All right, so I reviewed some research that might be important to consider for Lake Treman. So the paper that I reviewed was Meta-Community Theory Meets Restoration by Christopher Swan and Brian L. Brown. Meta-community theory considers local and regional effects and how varying degrees of isolation along a network, such as a river, can change these effects. So Swan and Brown used meta-community theory in their study of restored and unrestored streams to determine how local and regional effects can affect the success of restoration. So in order to determine the isolation of a particular point along a river network, they used the Strehler stream order method of classification. So this is just a little picture depicting the basics of Strehler stream order classification. So you can see that a first-order stream, two first-order streams come together to form a second-order stream, and two second-order streams come together to form a third-order stream. A first-order stream cannot come together with a second-order stream to make a third-order stream. It must be two second-order streams to become a third-order stream. So Swan and Brown found that in first and second order streams were classified as isolated headwaters, and third and fourth order streams were classified as main stem waters. So Swan and Brown found that in these well-connected main stem reaches, regional effects were more likely to play a role in biodiversity of benthic invertebrates, whereas in isolated headwaters, local effects were more likely to play a role in the biodiversity of benthic invertebrates. So what does this mean for Lake Treman? Lake Treman, or the stream that became Lake Treman, was determined to be a third-order stream. So what this indicates is that regional effects could potentially play a larger role in determining the biodiversity of our site than local effects. So the dam at Lake Treman could potentially be impeding species dispersal at our site. So if we potentially remove the dam, it could potentially increase the biodiversity of our site and the river around it. And one final thing that Swan and Brown made an effort to bring out in their study is that sites like Lake Treman should and should be researched to further Restoration Ecology's understanding of how local and regional effects affect restoration and how we can better restore sites like Lake Treman. And now we'll move on to Ryan and bacterial contamination.
So relating to the benthic diversity and overall human health, we wanted to investigate like certain types of contamination, such as eco-contamination. So to do that, we collected 6 samples along the watershed. We collected 6 samples along the watershed, upstream to downstream. And then we took them back to the lab, and to test for food contamination, we used this quanti-tray system, which is essentially trays for each sample. You fill it with 100 ml of a water sample, mix it with reagent, and incubate it for 24 hours. And then if the E. coli or any of the coliform bacteria we're looking for is present, it will interact with the reagent, and then it should glow under a black light. So that's the example. So these are some pictures from our collection from various sites. And this is all of the samples lined up. And as you can see, there's a gradient of contamination. So the ones at the end really glow. These are depositing as the sediment deposits the contamination. And this is a concern because, as you can see, the swimming areas where there's human interaction with the water is very contaminated, so that could be a possible concern for human health. And just like as a conclusion, this is sort of hinting that there needs to be more research done to where this contamination is coming from, and if it's like a human source or if it's from livestock. We really need to like locate and pinpoint these contamination points to prevent it in the future, to protect human health and just the overall health of the water system.
So during our project, we also did vegetation analysis, and we had 3 main objectives. We wanted to identify the diversity within and around the marsh. We wanted to identify the main cover types within the marsh, and we also wanted to identify where the invasive species are, because I know that's a big priority for the state parks workers. So the way we did this is we had 21 sites that we did a drone flyover, as you saw at the beginning with the video. And at each of these sites, we collected representative samples of soil and vegetation. And these are just a couple photos of us in the field doing these field sites. After we collected all these samples, we went up to the Bailey Hortorium and worked with Anna Stalter, who very kindly helped us identify all of our samples. We also did nested quadrats downstream of the marsh, and this showed us that there's about 33 species in a 6-meter squared radius, which defines the understory in the forest below the marsh. We also did variable tree radius plots, and we found a total of 11 tree species. This sounds pretty low, but it makes sense because they're woody species as opposed to herbaceous species, as we did in the nested quadrats. So from this, we calculated the % dominance for each species and the trees per acre for each species. So Parks cares about the most is probably invasive species. So we identified all the different regions where invasive species were found in the marsh. And we have Japanese knotweed, we have reed canary grass, and we have Phragmites. I'd like to point out that Phragmites is pretty centralized in a localized region, but the reed canary grass and Japanese knotweed are spreading throughout the marsh, which might be a big issue. So these are the 3 invasive species we're focusing on. Falopia japonica, which is the Japanese knotweed, Phragmites australis, which is Phragmites, and then Phalaris arundinacea, which is the reed canary grass. We also looked at the diversity in and throughout the marsh, as well as downstream. And when you first look at this marsh, you're like, it's just a bunch of cattails. What's going on in there? But in reality, we found 66 different genera of different plant species in there, and it actually equated out to about 90 species, and we still had some unknown, so it's probably closer to 100 species. Downstream in our nested quadrats, we had 33 species, as I previously mentioned, and 11 species in the variable tree radius plots. And I also would like to point out that as you're going from the inlet of the wetland down towards the dam, diversity is decreasing as you're going down, which, you know, might impact the vegetative impact on this field site. We also realized that some trees are starting to assist in succession of this marsh. So the tree species that we found are Platanus occidentalis, which is American sycamore, and Acer negundo, which is box elder. So most of the trees we found of Acer negundo were just little seedlings, but there were a couple mature trees, so we took a coring from one of these mature trees, and it aged out about 20 years. So this just demonstrates that succession is not an immediate process. It's not going to fix our problems as quickly as we would hope. So we might suggest planting some tree species to try and help stabilize the banks with the roots, or also shade out some of the invasives that I previously mentioned. Now, this is where it starts getting interesting. When we first visited our field site, we saw this big empty field of where there used to be Japanese knotweed. And we found out beavers actually had created a knotweed dam using all this stuff they had cut down. And, you know, that sounds pretty crazy, but it gets crazier. This knotweed that these beavers are cutting down are actually water rooting, and the beavers are acting as a vector. So this is them actively spreading an invasive species, which might have a larger impact not only on our site, but also throughout the county. So the state might want to look into this a bit more, because it's obviously a big problem. We also, when we were going through all our drone photos, we found this interesting thing happening. We found some carex, which is sedge, just in the middle of all these cattails. And it's kind of interesting. We're thinking this might be a native sedge, so it would be good if you plan on replanting in this marsh to use this, because it's obviously outcompeting typha, which is a large dominant in this field site, and it's also low growing, so it's not going to be taking over too many other habitats for other species. When we were doing our variable tree radius plots, we found a ground cedar, which is a club moss, and it's actually a state protected plant in New York State. So if the state needs some interesting aspect to, you know, really sell to whoever they're working for, like, this is an important site, we need to focus on this, here's one aspect you can go at. So in conclusion, we just want to recommend a couple things from the vegetation aspect. The state might want to do more fall and spring sampling of vegetation. We were very limited for this because we only had this fall semester to do this, but there's many spring ephemerals and other plants that I'm sure we missed. We also suggest doing more upstream nested quadrats and variable radius plots just to get a better understanding of what's going on. Definitely studying the impacts beavers are having on these invasives. Possibly getting more funding for the knotweed, and we believe it's coming from an upstream source, but we're not really sure. Using Watkins Glen as a reference site, we'll go into more detail about that very soon in this presentation, and possibly identifying some important wildlife habitat. We found some different species like wood ducks and blackbirds when we were doing our field work. So now I'm going to turn it over to Karen and Daniel for soil analysis.
Okay, last but not least, we're going to talk about the soil analysis that we did on this site. And we were trying to answer two primary questions. One being, are there any issues with the soil that can limit the plantings and recommendations we have? And also, if we were to remove sediment from the lake, is the dredge material hazardous? And if so, what can we do with it? And to answer these questions, we're looking at both surface soil collected around Lake Treman and upstream, and the dredge material that was mentioned earlier in this presentation. With surface soil, we looked at aggregate stability, texture, active carbon, and nutrients, and with the dredge material, we were looking at heavy metals and soluble salts. And our additional challenge was to do all these tests on a small class budget, but luckily the Cornell Soil and Health Lab was really generous and allowed us to do a lot of these tests ourselves for free, and then be strategic about what tests we submitted for them to do. We looked at 25 different heavy metals, some of which are shown in this chart with our 5 dredge samples and the list of heavy metals. And what we found is that the majority of the heavy metals are pretty far below the level of concern that New York State has, so that's good. There were slightly elevated levels of nickel and zinc. You can see the yellow highlighted parts there with the dredge sample 1 and 2. But both were within what we call the rural background soil level. That's what you might naturally find in upstate New York soils. And they were also pretty far below the level of concern for public health and aquatic health. So they are things to keep an eye for. And if you do plan on dredging or removing sediment, we would recommend taking more samples of sediment from the lake that could be dredge core samples or water samples, just to verify that these numbers do represent the heavy metal content in Lake Treman, and then also acknowledging that if you're removing the sediment and moving it elsewhere with a place with high zinc or nickel levels, then that could be a problem. But overall, we decided that the sediment was generally suitable for any uses, and that there's not any problems right now for public health or ecological health. And then here we just have a map of the relative concentrations of nickel sampled in Lake Treman. And we especially want to show this because this was pretty prevalent throughout all of the heavy metals, where you find the highest concentration closer to the dam, further downstream, kind of similar to what the methane team was talking about earlier. So that's probably where you would be removing sediment, but it's important to acknowledge that this is also where you might have a heavier concentration of heavy metals, because of the finer soils, and that's just where sediment is accumulating. When we were drying out our samples, we started to notice these crazy salt formations, so we were like, we're gonna look into soluble salts. But luckily all the samples were pretty non-saline or only very slightly saline, which means that the sediment should be generally safe for any plants or trees you would want to add. And then moving on to the surface soils, we want to look at aggregate stability. And so this is how well soil aggregates resist falling apart when they're hit by heavy rains. And you measure it by sifting out aggregates of a certain size, putting it on a sieve, and then putting it under this crazy machine, the Cornell Sprinkle Infiltrometer, which kind of simulates what a standard rainfall would be for 5 minutes. And then all the stuff you have left is your stable aggregates, and you use that to calculate your aggregate stability. And here we have a chart of our results, and a chart that was used to score those results. And overall, we found that our soils have pretty medium to high aggregate stability. These might be slightly overestimated because we had a heavy sand content, but in all, this suggests that there's a pretty strong resistance to soil loss during rain events. So this is pretty good for the site, good for plantings. And Daniel is going to talk about the rest of our soil analysis.
Set it behind. Behind. There you go.
Great, thank you. So as far as texture, many of the soils on this site scored similarly. Much of our sand content was up here between 90% and 60%. So a lot of very well-drained soils, heavy sand. Some of this can have to do with some of our sampling methodology. If we went back and had a chance to do this again, we would be taking much deeper samples to get at not just what's left on top, but some of the smaller particulates underneath. The active carbon, this is an interesting measure. So when you consider the rhizosphere, all of the bacteria that are down in the root zone trying to help the plants and trees establish, they need active carbon for functioning. And this test uses an interesting oxidative reaction to determine how much carbon, not just straight carbon there, but how much is actually going to be available for reacting. We found that intuitively, given the heavy sand content, there wasn't a whole lot of active carbon in these samples. The sites that showed the highest concentrations, 7, 8, 11, 18, these areas that either had standing water or areas that were prone to deposition during flooding events. Additionally, there's a gradient as one moves through the wetland. If we have it oriented here, there's the inlet over here on the left and the dam reservoir down here in the bottom. So again, as flow slows down, you're going to have finer particle deposition. And that will help facilitate increased active carbon. Again, soil health lab at Cornell, excellent tool for anybody with a home garden, the 1060 soil nutrient analysis. Generally, this is going to be used for agriculture problem solving. Do you have an individual mineral or element that your soil is lacking in? So we use this to try and figure, like, is there anything on site that is going to derail future planting events? Or are there pH concerns that might help us? Do we shift towards an ericaceous replanting scheme or something a little bit more alkaline loving? As far as the nutrient analysis, really not a whole lot to say. Nothing is too high, nothing is too low. The soil will be really facilitate new plant establishment. Looking at pH, so generally pH is going to have a set, like around 7 will be neutral. The soils that we have on site are just above 7, so we're going to be in that slightly alkaline range, so we're not going to be planting ericaceous things, mountain laurels, that kind of stuff. But generally there shouldn't be a big issue with species selection from a pH perspective. Additionally, we have the organic matter, again intuitively, high sand content. Well-drained soils, there's going to be a low organic matter. A lot of soils range from 1 to 6%. Usually one won't see these below 1% except for in desert soils. So that's sort of what's being created there is an interesting little microbiome where the sand or all the organic matter washes off of the sandy soils. Here we can see it again. These areas that have, well, 7, 8, and 11, this is a beaver dam area, and there's also flood deposition in that spot. So fairly high OM there. And then bringing it all together, what we tested of the dredge samples didn't throw back a lot of red flags. However, using an Ekman dredge, which is a claw that goes down and samples the top couple centimeters of soil, that lets us know recently what has happened. Now, what happened in the 60s or in the 50s during any kind of development in the area, we don't know. So one of the ways to get at that would be to do cores and test various layers of those cores. Beyond that, there are almost no concerns when it comes to plant establishment. Many things should do well on this site. There will be a gradient, of course, of closer to the waterway, sandier soils, you're going to want to have things that establish more easily. But it really shouldn't be a problem for the site. This is a great resource. It can be available digitally. We used the entire way through doing our standard operating procedures, through our interpretation of data. So whether at work or at home, if you need to do soil sampling, I suggest this resource. I'm an arborist at the Botanic Garden, and this is likely a self-indulgence, because if I don't talk about trees for one presentation, I've probably done something wrong. As far as replanting, there could be a lot of things accomplished by that. Some of it could be stream bank stabilization, like we're seeing these willows down in the bottom right. It could also be invasive competition strategies. A lot of the invasives on site obligate full sun organisms. If you can establish some kind of successional solar barrier above them, then that might be a mechanical-free, chemical-free method for treating it, but over a very long term. To that end, some of the species on site that are already doing that You can see here, this isn't Lake Treman, but it's what it could look like with a bunch of red stem dogwood. A lot of willows establishing on the banks. They're doing great. Willows and poplars especially, right in those sites. And then largest trees on site right now are the American sycamores. For some species diversity, I'm a big fan of deciduous conifers, taxodium. The bald cypress is very common in these kind of habitats, and they will outcompete many other woody species given the site constraints. If you want to get fun, I really liked the metasequoia. It was one of the most common wetland tree species on the North American continent up until about 2.5 million years ago. So, is it native, is it non-native? I don't know. But they do love this kind of site. Here we can see this is a stream, and these trees have roots right in the stream. So it could be an interesting woody addition to any kind of replanting strategy. If you consider that a lot of the downstream and current site draws are an element of tourism or an element of that ecological experience or novelty when it comes to grant writing. Another interesting native tree, Kentucky coffee tree, they have lost their evolutionary seed spreaders whenever the large arthropods, or excuse me, large herbivores left the continent. So you're only going to find this tree where its seeds distribute along streams. But again, very competitive in saturated soils. It would establish well. Additionally, it's a colony-forming tree. If you keep the beavers on site and they mow a tree like this down, it'll just come back from the roots. And yeah, final concern, the beavers, it's easy to say, yeah, just remove them, but they do a lot of the things that one might be trying to accomplish in a wetland maintenance or wetland restoration thing. They build dams, they slow flow, they retain sediment. They also eat all the trees that you try to plant. And as we've seen earlier, they do have potential vectoring of the knotweed. So in summary, we looked at as many things as we could on site. We tried to get the physiological characteristics, get the environmental issues, and looking at the biotic systems. One really important ecosystem service, clearly, is that there is sediment being retained. This is sediment that isn't going into buttermilk falls. This is sediment that isn't making it into the inlet or into the lake. So this is a really powerful regional benefit for a lot of shareholders. As far as invasives, there's always this dichotomy. Invasives bad, natives good. But when it comes to a large site restoration, we've seen these invasives do some potentially positive ecosystem services. The phalaris is retaining soil along banks while there is that woody transition. So do we immediately remove the phalaris, or do we focus on some of the other things like the phragmites? And again, should the beavers go? It would be really interesting to track some of those cut water-rooted sections of knotweed and see where they're ending up, and are they establishing on the bank as they get through the spillway. We have a little bit before big decisions have to be made, provided that the dam continues standing. Again, about 30 years before this will entirely silt up and we'll lose some of those sediment retention ecosystem services. On the other side, removal of the dam could be really important when it comes to arthropods or other benthic invertebrates who use that waterway to travel up and down. As far as gathering more information, there's a lot of resources locally to us. One that we advocate for and that we've referred to a couple times is the Watkins Gland site. They have a couple low-bed dams. One of them now is just a successional forest, and the other still has that wetland or lake feature. So it would be interesting to determine, here's what we want to see in the future for Lake Treman, and then go to these sites and see how those sites either accommodate or mitigate reaching those goals. And finally, I'll turn it over. Oh.
So we just want to thank all the people that have facilitated us completing our project. we couldn't do it alone. We needed the help of experts to kind of help us interpret and even do some of the things we've done in this project. So like, thank you all, and thank you guys for being here. And so we threw a lot of information at you, and you probably need some time to digest, but you'll have questions. So these are a few resources you can reach out to before we publish our report. So yeah, these are some cool references you can look at. And I'll turn it over to Audrey for final wrap-up.
All right, so thank you guys. And if you have any questions, now is the time. And we can direct it towards individual people, specific groups.
Well, I have been doing quite a bit of work on sediment transport, and especially along 6 Mile Creek. And I have some problems with just looking at off-stream sources of sediment coming into the lake and not looking at the possibility of in-stream sources in 6 Mile, and I think in some of the other creeks on here, but I haven't got the data. We've had a period of stream degradation for about the last 50 years. That means the stream has been cutting its way down, and that's because the land use has changed. I mean, originally when everything was forested around here, the relative input of sediment from the uplands, from off-stream sources, was low. And we had narrow channeled, wide, meandering streams, but when all it was deforested and turned into agriculture, you had tremendous input of off-stream sediment, which caused the streams to upgrade, fill in, and become multi-channel and all this kind of stuff. I mean, I didn't make that up, but people are quite aware of that. But, you know, the area's become reforested now. And so what does that mean? That means there's less sediment in general coming in from off-stream. So what's that mean for the stream? Well, the stream had tried to get an equilibrium with this tremendous amount of sediment coming in, which meant it had more energy. And now it has this energy, but it doesn't have any sediment, because there's a balance between energy and sediment. And so what's the stream doing? It's removing that sediment that has been stored in there. And 6 Mile, well, I live in Journal Crossroad, and I've been monitoring the stream channel there since just after Agnes, 1972, when we moved in. And this is, you're not going to believe this, but that stream channel has degraded about 14 feet over that period of time. They've stabilized it now so that the measurements make it good. So I think it's really important to get some measurements in upstream, in buttermilk upstream. Not in the gorge area where Comfort Road, Comfort and what's that other, anyway. That area, but up above that, the stream is out of the bedrock, and so there it's capable of being degraded. And it could very well be, like 6 Mile, that a very large fraction of the sediment that's coming into Lake Treman isn't from those. I'm not trying to defend agricultural procedures, but it isn't coming from those agricultural lands. It's coming from in-stream degradation. Yeah.
Can I respond to that?
Yeah.
So the point of identifying the erosion hotspots was basically based on the curve numbers in those areas, so addressing the permeability of soil in there. But the actual calculation for how much sediment is being deposited is just from elevation data. So it's just how high the sediment is now relative to what it was like before the dam was constructed.
Yeah, okay, that's two different things. You're going to measure how much sediment is being collected behind the dam.
Right.
Okay, and that's independent of where it came from. And all these soil measurements, and I get in an argument with Todd Walter sometimes about going about this, but it really doesn't do a good job on determining how much sediment's coming from the stream banks and stream degradation. You have to make some measurements. And you know, that takes time, but maybe there are some data out there. For instance, I don't know how many bridges go across, but the county engineers, at least in some bridges, always measure the depth to the channel or the water surface. I don't know. But there's some, they're not the best data, but there's some data there that could be used to determine whether or not there's significant degradation going on.
upstream.
Upstream.
Yeah, I think that's something very important to consider. Yeah.
I have a question for the invasive species team. You looked carefully at beaver and the potential impact that they may be having with Japanese knotweed. Did you also look for evidence of deer browsing and how that might be impacting the species distribution?
So we weren't looking too closely at deer interference, but we did, when we were in the field, we did see some deer prints on what we like to call Knotweed Island, that image I showed you where all the knotweed was cut down by the beavers. So there is maybe some potential for deers also browsing and interfering with some of the species composition within the marsh, but it's definitely something we should look more into in the future.
I know we've seen deer and deer droppings frequently as we've hiked that trail.
Another great thing that needs more research.
Just anecdotally, we have knotweed problem, and I don't give a damn how much beavers are exacerbating that problem, but it ain't going to stop. Every little piece of knotweed that comes downstream has the capability of rerouting, and if there's knotweed upstream, sooner or later it's going to be knotweed downstream.
That's what we're worried about.
Well, I think that, say, thinking about getting rid of beaver because they might be transporting knotweed is not a good, I mean, maybe you get rid of beaver for a different reason, but I don't think that's a reason.
Can I comment on that?
Yeah.
We looked a little bit into that, and I mentioned like maybe looking for sources upstream for the knotweed to try and also get rid of that problem, but just in addition to that, we're thinking, you know, trying to minimize the more spread of it with the beavers also present on the site.
I mean, we've been trying to get rid of knotweed. It's almost impossible.
Yeah, I mean, as is with any invasive species.
I mean, I certainly wish deer would eat more of it. They'll eat it in the springtime. They won't eat it in the summer.
Yes. It's definitely not an easy problem to fix, but, you know, just giving the state different options.
Find something that eats it.
There's another question back here, Audrey.
What's in the future of this? Is this just a class project, or is there something that's going to carry on after this is over with? Where is it going?
This is a class project, and like every time I've offered this class for the past 16 years, we take on a pro bono consulting project for a local government or nonprofit, and this fits into that context very well. Where it will go from here kind of remains to be seen. I'll suggest as an example, 5 years ago we looked at Spencer Lake, and Spencer Lake has a low-head dam, a shorter dam, not as tall as this dam. And the Corps of Engineers and State DEC wanted the dam removed. It dated from the 1820s. And they were assuming that Spencer Lake was 50 feet deep, and that could pretty much wipe out the downtown portion of Spencer had this dam failed. So we did a lot of bathymetry down there, and we found that yes, there are some ice melt pockets there that are 50 feet deep. But upstream of the dam, there is a subsurface ridge and only about 30 centimeters of water over that ridge. So should the dam break, for example, the likelihood of 50 feet of water times 29 acres blowing out the town of Spencer were relatively small. Well, they have been, the state was, people who came to this presentation, a presentation very like that, were very, they said, huh, who knew? They have still yet to figure out exactly what the proper course of action should be. And you're sitting next to a former student in this class who worked on the Spencer Lake project. And so as far as where it will go, this will inform state and local and perhaps even federal authorities on deciding where it should go, you know, where they would like it to go. But this is a not exactly a one-off thing, but it is a class project. And tomorrow's the last day of the semester, so we're not kissing the project goodbye. There will be a report, a technical report, that will hang out and continue to stimulate discussion and hopefully inform decisions. And yes, Jim.
Yeah, I would like to say, build off of that we would very much, as a representative of state parks, I think we got a ton out of this. We had worked with Tom and with the class to try to help inform a resource management plan for that area, which is important for us. And so we've got representatives from our environmental field team here, our planning, park operations, and invasive species, as well as educational departments. So there's stuff in here already that I think we can act on. That's small scale stuff that's going to have some impact. So we feel like we've got a little bit of a roadmap, a much greater understanding. And then as far as the dam goes, it also has some historical significance. Sure. As an artifact of the Civilian Conservation Corps. So we have several mandates that we're required to live up to, and having this information is going to really help us to do that. So we're looking forward to digging into it.
Yeah, great. So are there other questions or comments?
I just had a quick question for the class. Amongst all of you, did anybody strongly feel that it should be returned to a lake? Or were some of you like, no, it's going to stay a marsh? Like, was there any debate about where it should go or what it should be?
I think like the different groups were like prioritizing different aspects of the system. So like it performs like a variety of services, but like hydrologically, for instance, it would be nice if we could redirect flow with like a little more flexibility. But then like the invasives team probably found like trade-offs between like what a lake does versus what a marsh does.
Right.
But I think overall we think that retaining some aspect of the marsh is definitely in everyone's interest.
This, by the way, is another former student in this class who, thank you very much for coming. I appreciate it. So in terms of larger impact, these guys do go out and actually do things, and I'd like to think that in time and over the course of their professional careers, it does influence the way they approach problems and things like that. So, okay, thank you very much, everyone. And if you want to ask additional questions, I'm sure the students would be eager and available to answer those. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.